Okay, welcome back, everyone. Last series of lectures, or really the last lecture, we started talking about protein structure. There's a number of different levels of protein structure, and the first one was known as a protein's primary structure. If we look over here on the left-hand side, a primary structure was essentially a protein's amino acid sequence, importantly from N-terminus to C-terminus. So it is that amino acid sequence. And we found that within that amino acid sequence, we had some areas which were considered very ordered structure, as opposed to areas which would be considered random structure, as depicted here in this primary sequence. Ultimately, these areas of ordered structure were known as collectively as secondary structure. And we went over the two types of secondary structure over on the right-hand side, the alpha helix and the beta sheet. There are two depictions here of each of these types of secondary structure. So we're going to move on right now and start thinking about the next level of intricacy in protein structure. And it turns out, obviously, or to follow the cadence, that this will be called protein tertiary structure. Tertiary structure of a protein. So in a protein tertiary structure, overall, if we had to think about it in a general sense, we look up here at this first bullet point, it would be essentially its three-dimensional topography. It's essentially the shape of the protein. We are going to be looking at exclusively globular proteins. Globular proteins have a three-dimensional shape that is like a glob, essentially. If we look at some of these, which we will in a moment, they look like big globs. And that is in contrast to fibrous proteins. Remember, that was the other type of protein. We're not going to take a look at that type of tertiary structure, even though it exists, but we're going to focus only on globular protein structure. So we're talking about a shape. And if we think about it, the protein's primary structure, which you can think about, as a string or as a length of yarn, really, because it's a sequence of amino acids. And within that string of yarn, or that sequence of amino acids, we have areas of random structure, those random loops. We have areas of ordered structure, alpha helices perhaps, or beta sheets, and they ultimately all glom together. They collapse on itself, like the analogy of a string of yarn. If we took a long string of yarn and bashed it all up into a ball, well, we would have something that looked like a ball of yarn or a glob or something like that. So really, all of that primary structure of a protein with its areas of secondary structure collapses upon itself to form a defined three-dimensional shape. Now, importantly, we talk about the term defined here. It has to be a specific three-dimensional shape. So every copy of a protein which collapses into our globular protein, which collapses into a tertiary structure, every single copy of that glob that it forms has to look exactly the same. So if we look down here at bullet point 3, here's some representations of representative globular protein tertiary structures, and shockingly, they look like globs. Really, they're looking over here on the left-hand side. We see one way of representing a particular protein's globular protein's tertiary structure, and I should say that these representations of protein structure are derived from actual instrumentation through computer generation of an actual shape of a protein. It is not complete guesswork or just a random glob. It is the specific three-dimensional shape of a protein. If we look over here in this, and these are called space-filling models, over here on the left, we see a protein which really does look like a glob, but every single copy of this protein must have exactly the same shape. And importantly, the proper functioning of these globular proteins depend on this specific shape. Therefore, if we looked at on this structure, some of these protuberances, for instance, right here, or this shape right here, or these protuberances right here, if that were to happen, or if that were to differ at all in any of the copies of these proteins, we would have a situation where this particular protein would not be able to carry out its assigned function. This three-dimensional shape, this topography of these globular proteins must be exactly the same on every single copy, or the protein will not carry out its assigned function. The reason for that is so much of biochemistry involves protein-protein interactions, where one protein will fit like a jigsaw puzzle piece into another protein, and it has to be exact, or the two will not come together, will not properly convene together, and that signal transduction pathway, or that function required of that protein, breaks down. So because this three-dimensional structure of a protein is so important, and the maintenance of this three-dimensional structure, we have a number of active forces at work in any globular protein that work to maintain that tertiary structure. So we're going to talk about these four forces here which maintain the tertiary structure of a protein. The structure over on the left shows a ribbon, and that ribbon is essentially the protein's primary structure as it meanders around in a three-dimensional shape. Here's some area, here's an alpha helix perhaps, here's some random coil, here's another alpha helix, and it just sort of goes on its way, sort of in this meandering of a bashed up ball of yarn into a three-dimensional shape. We can think of it like that. But that meandering of that primary structure is crucial. We have to have the same three-dimensional shape. So once it is all bashed together, we have four forces which maintain its tertiary structure. We can think of these four forces as little staples that hold that three-dimensional shape together. So the first one that I want to talk about, and I'll talk about this a little more as we move along, are called hydrophobic interactions between side chains. Now, importantly, tertiary structure is these four forces, and the tertiary structure is maintained through specifically interactions between amino acid side chains, just the side chains. It has nothing to do with interactions between the components of the peptide backbone. You remember that in secondary structure, it was very important that the secondary structure, alpha helix and beta sheet, were all stabilized by interactions between components of the peptide backbone. Those hydrogen bondings. It had nothing to do with interactions of the amino acid side chains. Here in tertiary structure, that script is flipped. In fact, is so much so that the only interactions that these four forces participate in are between the amino acid side chains, nothing to do with the components of the peptide backbone, those hydrogen bondings. So getting back to what I was bringing up first are hydrophobic interactions between side chains. So if you remember, when we started looking at the side chains of amino acids, full 10 of our 20 amino acids were considered amino acids with a hydrophobic side chain. That means exclusively or primarily hydrocarbon. Hydrocarbon structure is very hydrophobic. It does not like to associate with water to the extent, and we saw this in the structure of membranes, in the structure of soap micelles, that hydrophobic areas within molecules will coalesce to exclude water. We've seen that twice. And the same can be said for these hydrophobic interactions between side chains. If you look over on the structure, hydrophobic interactions essentially mean interactions between hydrocarbon side chains. Here's perhaps a side chain of an alanine and a valine amino acid. Here is a couple of aromatic rings, perhaps from a couple of phenylalanines. Now, these associate strongly together because in the environment around a protein, that's all water. Remember, globular proteins dissolve in water, so there's water all around, but inside the protein it excludes water. Here in the interior of the globular protein, we have these hydrophobic interactions of our more hydrocarbon side chains inside, and they exclude water. It's an active effect, which we'll take a look at in just a moment. So one thing we can say is that hydrophobic amino acid side chains occur, basically situate their side chains towards the center of the protein. There's not a lot of hydrophobic side chains on the surface of the protein where we see all of the water that it's dissolved in, but just in the center, to the interior of that protein. So hydrophobic interactions sort of stand apart from many of the other four forces. The second force, which maintains tertiary structure, are hydrogen bonding between certain side chains of amino acids. We looked at, there was a grouping of our 20 amino acids, I think there's about 5 of them, that had what were called hydrophilic neutral side chains. These hydrophilic neutral side chains were ones that typically had a hydroxyl on them, OH, or NH bonds in an amide. There were 5 of them. You can look back at what they are, but we could imagine on some of these, so we get these hydrogen bondings occur between hydrogen bond donors and acceptors, but we've seen hydrogen bonding before, how it ultimately interacts, but these act also as staples at various points within this three-dimensional structure, which holds that three-dimensional structure together. These neutral hydrophilic side chains, essentially hydrogen bonders. The next level are called salt bridge, or rather ionic attractions between side chains. Remember, at physiological pH, some of our functional groups are ionized. For instance, amines are ionized as positively charged. Carboxylic acids are ionized as negatively charged carboxylates. So we have the possibility of opposite charges attracting one another of what is an ionic interaction, an ionic attraction, which is also known as a salt bridge. We see that right here. Salt bridge, with a salt are 2 ions, a positively charged ion and a negatively charged ion, and opposites attract. So here we have a positive negative interaction over here, which again is attractive in nature and maintains that tertiary structure. The last of the 4 forces which maintain tertiary structures are specific for only one amino acid, and that amino acid is known as cysteine. If you remember, if you look back at our structures of amino acids, cysteine was the one that had a thiol side chain, and thiols can form disulfides between the side chains of cysteines under oxidative conditions, which can be achieved within the cell or within the bloodstream or that sort of thing. We give rise to these disulfide bridges, shown suggestively here as just two sulfurs together. They must be cysteine side chains, and while it is a rarer form of these 4 forces, it does exist in many proteins as yet another staple which holds together the tertiary structure of a protein. That's so important to maintain that tertiary structure. Notably, disulfide bonds are the only interaction which is covalent in nature. Remember, these disulfide bridges are covalent linkages. I would like to go back and explore that first force which maintains tertiary structure now. Again, a little bit more, just one more slide, the hydrophobic interactions between side chains. If we look at it, this is called the hydrophobic effect in proteins, and if we look over at the structure on the left-hand side, we can follow this this primary structure, if you will, of this protein, this meandering of this peptide backbone, and within these amino acids we have different side chains. You know, here's a side chain here, and so on. These red and blue dots are considered side chains. Now, as we know, some of these side chains can be hydrophobic in nature, water-hating, and some can be hydrophilic in nature, water-loving. So when this protein here folds into its tertiary structure, we get something where we see perhaps over at the structure on the right, where now we're in our tertiary structure, but if you'll notice, we dispose all of the hydrophilic side chains outward on the surface of the protein towards the water that this globular protein is literally dissolved in. And more importantly, we see the red dots, which are representative of hydrophobic amino acid side chains, disposed towards the center of that protein. Importantly, they point themselves toward the center, and to such a degree that if that protein were to start to open up a little bit, it would allow water in to contact with these hydrophobic side chains, and the hydrophobic effect would take effect and exclude that water and reform that three-dimensional shape that was starting to unravel. It's a very active effect and one of the most important ones in proteins. It is important to mention here, remember back a few slides ago when I showed you those representations of tertiary structure based on a computer representation of actual X-ray diffraction data, instrumentation data from an actual protein crystal, we were able to get an idea of the topography, that three-dimensional topography of a protein and its tertiary structure. And the way I was showing it in the examples I showed you were mainly of the type of resolution as we see over on the left-hand side of this molecule right here. Sort of, we can see some general protuberances and shape, but a lot of it is left up to the imagination. So that was all sort of before 2012, and a lot has occurred between a lot of technological jumps have occurred between then and now, which now allow that the proteins to be visualized much more clearly. So over here in the present day, we can actually see these proteins where we get a much better idea of their secondary structure. See here some alpha helices over here they're very easy to see. And even if you look very carefully, the structures and the dispositions of the individual amino acid side chains where we weren't able to see that before. So a lot of the structures you see today that depict tertiary structure are really pre-2012, but a real jump in technology has allowed us to really get a very clear view into the interior and exterior of the three-dimensional structure of proteins. Once any protein has achieved its tertiary structure, in many, if not most cases, it is considered a completed whole protein, which can then go off and carry out its assigned function within the cell or within the bloodstream or so on. In some proteins, there is an additional level of complexity, only in some proteins, however, and that would be known as protein quaternary structure. quaternary structure. Remember, tertiary structure is the complete protein in probably a majority of proteins, but a minority of proteins need an additional level of intricacy before it can reach its active form. So we need to have a different level now of complexity, and that is brought on by up to this point, we've been talking about, up to the tertiary structure point, we've been talking about one molecule, one very large molecule. Protein is polymeric, polymers of amino acids, but it is one covalent molecule up through tertiary structure. Now, with the case of quaternary structure, this is where we get associations of different tertiary structures, different very large molecules coming together, and they associate together. That's an important word, associate, because it does not mean covalently bond together. There is no covalent bonds holding them together. It is an association. A good example of this would be the tertiary, or rather the globular protein hemoglobin, as a representative quaternary structure of a protein. Now, hemoglobin in its tertiary structure is not active. It cannot carry out its assigned function, which is to carry oxygen from the pulmonary system to our cells. It is an oxygen carrier, but it is not active unless 4 units of hemoglobin can associate together. Now, in this drawing right here, we have 4 different colors, if you will, of hemoglobin tertiary structure, and they are now associating together. And when they do associate together, they are now considered in their active form, and they can do what one of its species, one tertiary structure of hemoglobin, could not do. It has to have 4 of them together in order to carry out its assigned function. Now, because it is not, the quaternary structure is not covalent in nature, we have to ask, well, what is holding these associations of tertiary structures together? Well, ultimately, it is the same 4 forces that maintain tertiary structure. Remember, the majority of those were associative attractions, like ionic attractions, hydrogen bonding attractions, hydrophobic attractions. They weren't covalent in nature. The exception to that would be covalent disulfide bonds, which don't usually occur in protein quaternary structure. It's really those first 3. But if we look over here on hemoglobin, we have these associations perhaps happening between these that are hydrophobic in nature. Maybe there's some hydrogen bonding side chains. Remember, we're always dealing with side chains of amino acids and these associative in these associations that hold together these different covalent structures to form a quaternary structure. Again, quaternary structure does not happen in every protein, or is not necessary in every protein to achieve its active form, but just in some proteins. Having gone through quaternary structure of proteins, that finishes us off for this very light, very nerfed up version or explanation of protein structure and, to a certain extent, protein function. Now, if you remember, I had told you that it is really a fool's errand to try and express the importance of proteins as a biochemical structure through one unit of study or a series of slides. But what we did was just to give a general idea of how proteins are put together, and somewhat of their function is what we tried to do. Now, in the very beginning of the proteins chapter, we looked at various types of globular proteins, various examples. One of these were enzymes. And enzymes enzymes are so important that we devote a unit of study specifically for this special class of protein structure. So here we are now in Unit 10, and we're going to start talking about this special class of proteins known as enzymes. If we had to put a definition to it, enzymes are biological catalysts. As we saw throughout the organic chemistry portion of the course, we saw that many, if not most, chemical or organic reactions required certain conditions, specifically catalysts, to carry out the reaction. We needed metal catalysts for hydrogenation. We needed catalytic acidic conditions for many of our particular other reactions. We needed heat sometimes as a condition. None of these things can happen in biological systems. They happen fine in a test tube, but they cannot happen within a biological system. If we had to acidify our bloodstream just to mediate a particular reaction or catalyze a reaction, any biological system that happened to would die. We could not inject say palladium metal into our systems, because any hydrogenation which required it, that would be toxic conditions. However, these reactions which require catalytic acid, which require the other types of catalysis, still happen within our systems, animal systems, vegetable systems. They still happen. So in place of all of those catalysts or catalytic conditions, is replaced with enzymes. Enzymes are globular proteins, biological catalysts. They catalyze the same types of reactions, but via through being a globular protein, and through some types of ways we're not going to be getting completely into. They carry out the same function, say, as acidification or other types of conditions. So enzyme, when I'm going to be looking at talking about enzymes in particular, but really this is the case with all catalysts. Enzymes are simply a subset of all catalysts. Catalysts in general do not change the equilibrium of a reaction. They don't change that. They do not change the energy level between reactants and products. If we look at the structures, the graphs down below, that energy level difference between the sum of the chemical energy within the reactants versus the chemical energy within the products, that doesn't change. What does change, however, with any catalyst, notably enzymes, since that's what we're talking about, is that we increase the rate of the reaction. And the way we increase the rate of the reaction is that we lower the activation energy of a reaction. So over on the left-hand side here, we see an uncatalyzed reaction with an activation energy which looks like this. If we catalyze that reaction by an enzyme or by any other catalyst, this could refer to a biochemical reaction or a reaction in a test tube. But if we catalyze it, what that does is simply lowers that activation energy and allows for the rate of the reaction to increase. That's the only thing it does. So the talking point here is that we do not change the equilibrium of the reaction or the energy level difference between reactants and products, but rather the rate of the reaction through lowering of that activation energy. With enzymes in particular, we typically see a rate enhancement on the order of 1 million to 10 million times. So that's significant. In biochemical reactions, we essentially need these products now. We don't have time to wait for a long process. Most of these biochemically catalyzed reactions occur what would appear to be instantaneously, and they do need to appear to be instantaneous in order to satisfy the continuing needs of activity within the cell or other biochemical activity, but the general rule would be that rate increases are about 1 million to 10 million times faster than the uncatalyzed reaction. Importantly, enzymes themselves as catalysts, these differ from most organic catalysts in that one enzyme, not all the time, but most of the time, will only recognize one particular substrate. That would be, that substrate would mean a starting material in a reaction. Only one. Whereas, say, palladium as a catalyst for a hydrogenation will recognize any carbon-carbon double bond and catalyze its reduction to a carbon-carbon single bond, but one enzyme only recognizes one molecule, typically of a particular substrate. So if we think about this now, of the hundreds of thousands, if not millions, of different reactions that are going on in a representative cell in your body at any given time, there is a globular protein, an enzyme, for each one of those which matches it. It really speaks to the incredible complexity, just one part of the incredible complexity of life's processes within and outside of the cell. Now, we look at nomenclature in biochemical systems quite different than we looked at nomenclature in the UPAC nomenclature in organic systems. Nomenclature in biochemical systems is typically a little bit more colloquial in nature, but there are certain things that hold certain types of biochemical structures together. Notably with enzymes, any name that an enzyme has will almost virtually always have the suffix ASE, ACE, in identifying whatever functional class we're talking about. If it ends with ACE, it is almost certainly an enzyme's name. And that is similar to when we were looking at carbohydrates. If it ends with OSE, OSE, it is probably some type of carbohydrate, whether it's a monosaccharide, disaccharide, or polysaccharide. It is probably going to be, almost certainly going to be, a carbohydrate. But ACE defines the functional class. Now, with regards to the rest of the names of enzymes, I'm convinced that whoever comes up with enzyme names has to have a sense of humor, because some of these are very funny and very obvious in the types of either the types of substrate they're specific for or the types of function that they carry out. The name of an enzyme typically gives some hint as to its function, and so a lot of the time that can be guessed. So let's take a look at some examples of how we could take the name of an enzyme and deduce what its function might be. Here's a representative reaction, one that we've seen before, actually. Remember when we were taking amylose over here on the left, and through digestion in the stomach, that can form maltose disaccharide. It says essentially we're taking little snips, two monosaccharide snips at a time of amylose and forming maltose. It's the beginning of our breakdown of dietary starches until we eventually get to free glucose, which can then be transported to the cells and used to power or to extract energy from that fuel molecule and used to power cellular processes. But this is the first process. So if we look at this, look at the enzyme, and frequently that's given over the arrow here. Amylase. Amylase is now as the name of an enzyme gives us a hint as to its preferred substrate, that of amylose over on the left. Amylose is one of those dietary starches we had. So amylase tells us this enzyme is working on amylose, if you will. The next one. The term lipase, that is the name of an enzyme. Here's lipase right here over an arrow. Now, what might lipase be specific for? What type of substrate? Well, if we look at the first three letters in lipase, L-I-P, that's the first three letters in the word lipid, L-I-P. Now, a lipid, as we see here, is this neutral glyceride, a triglyceride, which is essentially fat structure, which we see in fatty tissue within our bodies, or it could be vegetable oil, it could be animal fats, it could be lard, it could be anything like that. It is lipid in structure, and that lipid structure, that triglyceride, needs to be hydrolyzed. As we see over here on the reactant side, we have water right there. And through the action of lipase, increases that reaction so that we get our fatty acids over here in the product profile. Those fatty acids, ultimately, as we'll see in the future, are going to be used as another type of fuel molecule. We'll see, in addition to glucose, from which we can extract chemical energy. But this is the first process in turning stored energy in the form of triglycerides, breaking it down into the representative fatty acids, and then used within the cells to extract energy. Lipase for lipid, if you will. Our last example here, the name speaks more towards the function of the enzyme as opposed to the actual the substrate for it. So this enzyme right here, known as a decarboxylase enzyme, remove carboxylic acid. So it removes a carboxylic acid. We can deduce that from that hint of a name. It catalyzes the removal of a carboxylate or a carboxylic acid from a substrate. On this particular reaction here in blue is given the carboxylate. COO minus is a way of expressing a carboxylate in a condensed structure through the action of decarboxylase. Well, it's not there anymore. It's just a CH2. So a carboxylate, COO, when we lose it, almost always gets blown off as a molecule of carbon dioxide. It has one carbon and two oxygens. We're going to see that over and over when we talk about energy transduction pathways a few units of study hence. But just for the sake of what we're looking at here, in this case, the name gives some hint as to the enzyme's function. The remainder of this lecture is going to be looking at a classification system of enzymes in which we're not going to be necessarily looking at the names of enzymes, but rather talking about names of classes of enzymes. These classes are the categories that we put enzymes into based upon the types of reactions that they catalyze. So we're going to be looking at this throughout the next few units of study. If we were to ask, say, the question of what type or what class of enzymes might catalyze this reaction, well, we'd have to look at the reaction, look at the reactant, look at the product, and say what has changed going from reactant to product. And based upon that structural change, we can think about what class of enzymes might have mediated that. So here we see the 6 different classes, 6 at least for now, of the 6 different classes of enzymes. The first is an oxidoreductase. Again, sometimes these can give some indication as to the types of reactions they catalyzed. Oxidize, reduct, or reduce. So it would catalyze oxidation or reduction reactions. Most of these classes of enzymes do have that hint to them, not all of them, but have that hint to them as to their function. So to go back down the list here, a transferase class of enzymes. We'll look at specific examples of these as we move along to the end of this lecture. Hydrolase, well, probably involves water. Lyase, well, lyase is going to be one of the ones we're going to find that does not have a hint to its function or its substrate given in its name. That is in the minority. Isomerize. Well, isomerase. I wonder what that does. Ligase. We'll talk about the action of a ligase as we move along. So on the slides to come, we're going to see example reactions of each of these different classes of enzymes, the enzymes which mediate these reactions. And we're going to try and look again at the starting material versus the product, and try and work out why, say, an oxidoreductase would be the type of enzyme that would mediate this reaction, or an isomerase, or so on and so forth. So let's look at the first one, that of oxidoreductase. We'll look at an example of that. So as the name implies, the oxidoreductase class of enzymes, they catalyze oxidation and reduction reactions. Much of the time we see that, or all of the time really, that oxidations and reductions are simple reversals of one another. And since all chemical reactions or organic transformations are in essence reversible, these oxidation and reductions are typically classified as redox in the same sort of breath. RED for reduction, OX for oxidation, redox reactions. That means either an oxidation reaction or a reduction reaction. And here now we have an example. Let's look at this example, and we'll specifically try to go from reactant to product and try to figure out what is going on. And I've drawn in red here where the action is on the molecule. But if we looked at this, we have this type of, the name of this particular enzyme here is called lactate dehydrogenase. Well, from that name, we can get at least its preferred substrate, lactate. Dehydrogenase, we'll look at that in just a moment. So look over here on the molecule. This hydroxyl is what is changing to a carbonyl. Well, it looks like we've lost a CH bond and gained a CO bond going from reactant to product. So that is diagnostic for an oxidation. So this must be, lactate dehydrogenase must be of an oxidoreductase class of enzymes. So that's the way that we can look at it. Specifically, if we look at the name of this again, the second word in the name, dehydrogenase, still has some indication as to its preferred function. Dehydrogen, we're losing hydrogen. If we look over at the structure here on the left, well, we are losing a CH bond. Dehydrogenating. We don't have that anymore over on the structure on the right. So we could say that still this indication, if you will, of what types of structure or structural change is still baked into this particular class of enzymes. Next class, transferase. Transferase. So a transferase class of enzymes catalyze functional group transfer reactions from one molecule to another. Let's look at this particular reaction. We have here alanine. This was one of our 20 amino acids. It's actually written here not usually the way we write these amino acids, that N-terminus to C-terminus, but again, in the form of a Fisher projection, we saw that sometimes in order to determine with the D or L configuration on amino acids. In any event, what happens here is in red. Let me clean up the structure a little bit here. In red, we have a transfer of functional groups. This amino group and this carbonyl group, if we look over to the product profile, are now transferred. One is transferred to the other. It doesn't matter what this specific reaction is, but we could look at this reaction and say, it looks like we have a functional group transfer from at least one molecule to another. Therefore, alanine transaminase, notice it has the word trans in it for transferase. Alanine transaminase would be of the transferase class of enzymes. The third class of enzymes, known as a hydrolase class. As it would be expected, hydro might be thinking about water, and what it does is these classes of enzymes catalyze hydrolysis reactions. Remember hydrolysis, hydrolysis, where hydro for water, lysis means to break apart or to cut. We're using water to break something apart. And in this case, we have a small molecule known as acetylcholine and water. We're using water to cut right there on that on that molecule, and we get two types of products where that water was then used to cut this molecule in half. Hydrolase. If you remember our hydrolysis of esters, that happens in the test tube with an acid catalyst. Well, that can happen in the body as well. We always have to hydrolyze esters for many functions within the cell, so a hydrolase class of enzymes would carry this out. The fourth class of enzymes. Fourth class of enzymes is known as the lyase class. This is an exception to the rule, wherein, at least as far as I can tell or I know, there is no hint in the term lyase as a class of enzymes which will delineate what type of function this enzyme class carries out. So sometimes that happens. In this case, a lyase class of enzymes will catalyze the addition of water over a carbon-carbon double bond. And when that happens, we know from organic chemistry, we get an alcohol as the product. So here over on the structure on the left, we have a carbon-carbon double bond here. And through using water adds over that carbon-carbon double bond so that we get H on one side, OH on the other. What we have created is an alcohol functional group. So again, if we looked at this starting material and then looked at the product and we thought to ourselves, what sort of enzyme, what class of enzyme might carry this out? It would be a lyase class of enzymes. The fifth enzyme class, known as an isomerase class of enzymes. Now we're back on the wagon of having the name of the class of enzyme predict what kind of function or what kind of reaction this enzyme class will catalyze. As isomerase class of enzymes catalyzes the structural rearrangement of a substrate to form a constitutional isomer. Constitutional isomers are the same molecular formula, but different atom connectivity. So that we get, again, we have to analyze both the starting material and the product and say, what has changed? Well, in this case, I've done it in red. Look, we have on the structure on the left, we have an alcohol functional group up top and a carbonyl, specifically a ketone functional group right below. On the product side, look, they are swapped. We have created a constitutional isomer. If we look at the name of this enzyme, isomerase, in the name gives us an indication of the class of enzyme which would carry it out. But again, it's most importantly, we're looking at the difference in structure between the starting material and the product to deduce the type of class of enzyme that mediated that reaction. Our last class of enzymes, the ligase class. Now, ligase, if we think of it, what might be an English derivative word for ligase, when we ligate something is ligated, we are connecting it or tying it to something else, like a ligament ties our bones together, or it ties a muscle to a bone. When you ligate something, you're tying it together. So ultimately, what a ligase class of enzymes does is it catalyzes covalent bond formation reactions with different molecules, which structurally connect separate molecules together. So in this case, we have one starting material, two starting materials over here on the left. So we have two starting materials. Now we only have one product where that nitrogen, if you will, has been ligated to the original molecule. So we've ligated two of these things together into one. So that would be an example of a ligase class of enzymes. Now, so the real question is, so what sorts of questions might you be asked on this, on a test or a quiz or an exam? Well, we could look at a couple of these. So we have a couple of different reactions here, and we could ask which class of enzymes catalyzes the following reactions. Let's see. On this top one, going from reactant to product, while a lot of the structure remains the same, I can pick out we have what once was an alcohol, OH here, becomes a carbonyl group, C double bond O. That is by definition an oxidation because we have gained a CO bond and lost a CH bond. So the class of enzyme which would mediate this would be an oxidoreductase class of enzymes. The bottom one, going from reactant to product, what we see here is everything is the same except in the reactant we have this carbon-carbon double bond. And between the two carbons on the product profile right here, we've left out a hydrogen right there because this is done in line drawing, but we can see or we can deduce that water was added over this carbon-carbon double bond to form this alcohol right here. And the class of enzyme that would mediate this would be a lyase class of enzymes, because we recall that a lyase catalyzes the addition of water over a carbon-carbon double bond.